So friends, we were discussing about the factors that can affect the efficiency of enzyme assisted extraction process, and in that we were discussing about the effect of enzymatic mixtures that are being used in this process. Now, since it has been seen that because of the multi-component nature of the plant matrix or cell wall, it is always desirable to use a mixture of enzyme, and therefore and these mixtures are sometimes also known as cocktail enzymes, and therefore nowadays commercially there are various mixture of different enzymes that are available in the market. So I have taken an example of 3 different commercially available enzymatic mixtures. As you can see, these are viscozyme, which is nothing but a mixture of 5 different enzymes: arabinase, cellulase, beta glucanase, hemicellulase and xylanase. Another commercially available mixture is lalzyme, which is mixture of pectinase, endocellulase and galactanase. And the third one is rapidase, which is a mixture of pectinase and hemicellulase. So there are numerous other mixtures that are available commercially that can be used in this process. Now we move on to another factor, that is extraction time and the concentration of the enzymatic solution or the enzymatic mixture that is being used under this process. By and large, it has been observed that the extraction time and the concentration of enzymatic mixture are inversely proportional to each other, though this rule is not universally applied. So if we say inversely proportional, it means if we increase the overall extraction duration or extraction time, then we will require lesser concentrated enzymatic solution, and vice versa. If we are using highly concentrated enzymatic solution, then we will be requiring a smaller or the shorter extraction time, because the concentrated enzymatic mixture will hydrolyze the plant matrix and the extraction process will get fastened up. In a study, it was found that maximum yield of orthodiphenols and total phenolic content from olive oil was extracted when there was a prolonged malaxation as well as high concentration of enzymatic mixture was used, and that mixture was made up of pectinase, cellulose and hemicellulose. And that is why I said that the inversely proportional relationship extraction time and concentration is not universally applied, but by and large we have seen this kind of relationship. Now malaxation time is nothing but malaxation is a process wherein we rub the fruit in order to make it tender, to make it soft, so that extraction process can be fastened up. Numerous other examples have been seen wherein the prolonged extraction time, they also decrease the yield, and this decrease in the yield of the active constituent or the desirable constituent has been largely found due to the degradation or oxidative degradation of the compounds, and this we have seen in almost all other extraction techniques which we have discussed. So if we increase the extraction time beyond a specific limit, then obviously that increased extraction time has a derogatory impact on the compounds that we want to extract. In another study, it was found that on increasing the extraction time of muscadine, it was found the polyphenolic compound or the yield of the polyphenol was declined. This was largely due to the thermal destabilization of those polyphenolic compounds. Similarly, the anthocyanin yield from the saffron decreased when the extraction time was increased over 3 hours and the concentration of the enzymatic mixture was increased above 5%. So here you can clearly see the impact of the concentration of the enzyme and also the impact of different enzymes on the overall yield. So what they did was they extract saffron by using 2 different enzymes, Cellubrix and Pectinix, and the effect of the enzymatic activity was assessed on the basis of the anthocyanin content that was extracted. They also studied the effect of the increase in the concentration of the enzymatic solution. So we can see in the graph, the white colored squares exhibit the effect of Celebrex, whereas the black colored squares exhibit the impact of pectinase. So here you can see that as we increase the concentration of Celebrex, there was hardly any impact on the concentration of anthocyanins that were extracted. This is one aspect. Second, if we compare it with Pectinex, you can see that the amount of anthocyanins that were extracted by Pectinex were almost 60% higher in comparison to that of Celebrex. So here is the impact on the extraction yield on the basis of the type of the enzyme. Further, if we further look into this graph, you will see that as we increase the concentration of enzyme solution above 5%, then the yield of the anthocyanins that has been extracted decreases slightly. So one can clearly understand through this graph that the specificity of the enzyme, because it has been found that the grape skin is rich in pectin, and therefore it is more susceptible towards the action of pectinase, and that is why pectinase is able to extract out large amount of anthocyanins. So this is the impact of the enzymatic concentration. Now we move on to another factor, that is extraction temperature. In all the extraction techniques, we have seen that increase in temperature has a positive impact on the overall extraction yield, but up to a specific value, because the increase in temperature reduces the viscosity of the solvent, which increases the penetrability as well as the solubility power of the solvent, thereby increasing the extraction efficiency. Increase in temperature in case of enzyme assisted extraction is important because enzymes are proteins and they exhibit optimum activity at a optimum temperature value. And if we further increase the temperature beyond that optimum value, it may cause denaturation of those proteinaceous molecules, which we call them as enzymes. But by and large, the enzyme of activity increases with the temperature, but again up to a specific limit. Higher temperatures beyond the critical values have shown to alter the three-dimensional structure of the enzymes, which are nothing but the proteins. And since their structure is altered, the enzymatic activity, that is the hydrolytic activity of enzymes, decreases, thereby causing decrease in the cell wall hydrolysis. And this increase in temperature also causes thermal degradation of the compounds, which will have a negative impact on the overall extraction yield. Again, various studies were done in order to study the impact, to see the impact of increase in temperature, and it was found in one of the studies that organosulfur compound content, when they were extracted from garlic, was reduced once the temperature was increased from 40 to 60 degrees during extraction as well as during storage. Increased temperature enhanced the mass transfer rate and solubility of extracted anthocyanins from Prunus nepalensis fruits. Again, beyond the when the temperature was increased beyond the critical value, the amount of the compounds as well as the extract yield was decreased because of the enzymatic denaturation. Now we move on to another factor, that is pH of the media. As we discussed, the proteins are nothing but the protein molecules, so pH has a major impact on the enzymatic activity. One has to maintain an optimum pH condition in order to exhibit maximum activity of the enzymes. One has to maintain pH in such a value that it should not interfere the enzymatic activity and should not be in the range of isoelectric point of those enzymes, because if we keep the pH in the isoelectric range, this may cause precipitation of the enzymes, and this may cause complete loss of the hydrolytic activity of the enzyme, thereby having a negative impact on the overall extraction process. but it has been observed, by and large it has been seen, that acidic pH favors the enzymatic activity, because this acidic pH destabilizes the hydrogen bond, thereby increasing the cell wall plasticity, which makes them more vulnerable towards enzymatic hydrolysis. This acidic pH also causes modification in structural protein configuration, again making the cell wall vulnerable towards hydrolysis, and it enhances the binding capacity of the enzymes on the cell wall, which eventually leads to the better activity. Again, various studies showed that the activity of enzymatic combination of pectinase and cellulase, which was used to extract taxifolin, was improved tremendously in the pH values of 5 to 6.5, and if we further increase the pH, it was found that it does not have any impact on the enzymatic activity. Similarly, the extraction of leucinin and epigenin from Cajanus leaves was improved with pectinase when the pH was in the range of 3.5 to 4, and when the pH was increased, there was a decrease in the enzymatic activity. Now we move on to the hyphenations of enzyme assisted extraction. As I discussed, that the beauty of this technique is it can be used in combination with any of the extraction technique, and on that basis we have different hyphenations of enzyme assisted extraction. We have enzyme assisted ultrasound extraction, we have enzyme assisted pressurized solvent extraction, we have enzyme assisted ionic liquid extraction, enzyme assisted microwave extraction, enzyme assisted supercritical fluid extraction, and enzyme assisted three-phase partitioning extraction. These are the few examples of modern extraction techniques. Now I will discuss how these hyphenations have a positive impact on the overall extraction process. So first is the enzyme assisted ultrasound extraction. So there was a study wherein the impact of different extraction techniques was done on the overall hydroxysanchule content that was extracted from Xanthoxylum species. As you can see in the graph, the first bar shows the content of hydroxysanchule when it was extracted by normal conventional solvent extraction, that is 4.12%. Then the fruits were extracted by only enzyme assisted extraction and then by ultrasound assisted extraction. If we compare the results of enzyme assisted extraction and ultrasound assisted extraction, we will see there is hardly any difference in the hydroxysanchule content that was extracted. But when we mix the two techniques, and in this mixing of the techniques, the fruits were first incubated with the enzymatic solution and then they were exposed to ultrasound assisted extraction, and you can see there is a tremendous increase in the hydroxysanchule content that reached to almost 6.5%. So this is the impact of the hyphenation of the two techniques. Here you can see the impact of various extraction techniques on the surface of the fruit. So the image A shows the impact of solvent extraction, the image B shows the impact of enzyme assisted extraction, the third image C shows the impact of ultrasound assisted extraction on the cell wall integrity, and the fourth image shows the impact of the combination of ultrasound and enzyme assisted extraction on the cell wall integrity of the fruits. You can clearly see that when ultrasound assisted extraction is used in with combination with enzyme assisted extraction, the cell wall integrity was completely damaged, and this is the reason why the extraction yield of hydroxysanchule was much higher in comparison to those when the other extraction techniques were used. Another hyphenation is enzyme assisted extraction along with pressurized solvent or pressurized liquid extraction. In this study, non-extractable phenolic content were determined when the plant material of sweet cherry was extracted by different extraction techniques such as conventional solvent extraction, pressurized liquid extraction, and pressurized liquid along with enzyme assisted extraction. we can clearly see in this graph that the non-extractable phenolic content was very less in comparison in conventional extraction method. It slightly increased in pressurized liquid extraction, but it tremendously increased when combination of pressurized liquid extraction was used with enzyme assisted extraction. Now, to understand this significant increase in the extraction yield, we have to first understand what are non-extractable phenolics. Now, I'll again take you back to the structural image of the cell wall which we discussed yesterday, and in that we saw that there are certain phenolic compounds which are bind to the components of the cell wall. Either they are bound to cellulose, they are bound to hemicellulose, or to lignin, or to pectin. Such bound phenolic compounds are not extracted by the conventional or the modern extraction techniques efficiently. And such phenolic compounds we call them as non-extractable phenolics. And these compounds can only be extracted once the cell wall is disintegrated. And this is the reason why when the combination of pressurized liquid and enzyme assisted extraction technique was used, there was a tremendous increase in overall non-extractable phenolic content. Another hyphenation is extraction by using combination of enzyme assisted with ionic liquid extraction. And we know that ionic liquids are nothing but these are the solvents that are composed of bulky organic cations and inorganic or organic anions, and these combination or these liquids are liquid at room temperature. There are certain advantages of ionic liquids to be used as solvents because they have a very negligible vapor pressure. They have good thermal stability, you can use at higher temperatures. Their viscosity is tunable because viscosity has a major impact in overall extraction efficiency. We can always increase or decrease their viscosity depending upon our requirement. These solvents are miscible with both water as well as organic solvents. So you can extract hydrophilic as well as hydrophobic compounds with these solvents, and they have good solubility and extractability for different organic compounds because they have variability in their polarity. Now, in this image you can see the impact on the overall chlorogenic acid content that was extracted. So, different aspects were covered in this study. In the first graph you will see the impact when ionic liquids were used with different enzymes, and you can see that the mixture of ionic liquid with pectin-X showed maximum content of chlorogenic acid that was extracted. In the second image you will see that in the first image different types of ionic liquids were used, whereas in second image different enzymes were used along with ionic liquids, and we will see that ionic liquids were used for extraction. They showed better results in comparison to the cationic liquids, whereas in case of different enzymes, as I already told, pectin-X or pectinase enzyme showed maximum extraction of chlorogenic acid. And in the third image you will see the impact of the mixture of ionic liquid along with pectinase as different concentrations. So here we cover all the three aspects, impact of all the three aspects, that is the type of the ionic liquid, the type of the enzyme, and the concentration of the enzyme in the ionic liquid has a great impact on the extraction efficiency of chlorogenic acid. Here is the effect of the combination of ionic liquid with pectinase on the surface morphology of cytomorphology of the fruits from which chlorogenic acid was extracted. So the image A is of the untreated sample. You can clearly see the structural integrity of the cell wall of the fruits. The second image shows when it was only treated with pectinase. There is a slight distortion in the structural integrity. Third image shows when it was treated only with the anionic ionic liquid. Again, structural integrity was slightly distorted. And the fourth image shows when the combination of pectinase and ionic liquid was used. You can see there is a drastic difference in the structural integrity. That is the reason why the chlorogenic acid yield was highest when the combination of the two was used. Another example of hyphenation is enzyme assisted extraction with microwave extraction. Again, the plant material is first incubated with the enzyme and then it is extracted with the microwaves. And you can clearly see the amount of the oil extracted when the combination of enzyme and microwave extraction is used is much higher in comparison to the oil content that was obtained when it was extracted only by using microwave assisted extraction and when it was extracted by simple hydrodistillation. And it's not only the yield, but the time of extraction was much lower in comparison when the combination of the two techniques was used in comparison to the conventional hydrodistillation. This is the effect on the surface morphology of the fruits. Again, this image A is of the untreated sample, B is of the conventional hydrodistillation, C is when it was subjected to microwave assisted hydrodistillation, and fourth one is the combination of enzyme assisted and microwave assisted hydrodistillation. And in the fourth image you can clearly see the complete distortion of the cell wall structure, which leads to the enhanced extraction yield. Again, the impact of the combination of enzyme assisted and microwave assisted extraction on the total phenolic content, and you can see the significant increase in the total phenolic content that were extracted when the combination of the two techniques was used in comparison to the conventional solvent extraction and when the plant material was extracted individually by enzyme assisted and microwave assisted extraction. There is another hyphenation when enzyme assisted extraction is used with supercritical fluid extraction. In a study, the impact of different extraction techniques, that is conventional extraction technique, that is maceration, modern extraction technique, that is supercritical fluid extraction, and the hyphenation of enzyme assisted and supercritical fluid extraction was assessed on the phenolic compound content like caffeic acid, paracumaric acid and synapic acid. You can clearly see that when we use the combination of the two techniques, there is a significant increase in the content of the phenolic compounds that was extracted. So the bottom line is, whenever we are using the combination of two extraction techniques, enzyme assisted as well as any of the modern technique, the combination or the hyphenation has shown a significant increase in the overall extraction yield and also the yield of the desirable components. Now we move to the last part of this lecture, that is the applications of enzyme assisted extraction. This technique is largely used to extract out the secondary metabolites from plants and more importantly from the plant waste, because there are the constituents which remain in the plant waste which have not been extracted by modern or the conventional extraction techniques that can be extracted by the enzyme assisted extraction. And the common example of such secondary metabolites are the non-extractable phenolics. So one can extract polyphenols, carotenoids, flavonoids, oil, pectins and anthocyanins by using this technique. This technique is also used to prepare plant-based drinks or fermented drinks from different grains. These enzymes basically help in enhancing the fermentation by hydrolyzing the cell walls, by changing the chemistry of the cellular structures, which leads to enhancement of the fermentation rates, and that leads to the production or help in producing different types of plant-based drinks, fermented drinks which can be used as probiotics or prebiotics. Now this technique is also used in food industry specifically to extract different types of flavors and colors from different plants, and like lycopene from tomato, carotenoids from capsicum or pumpkin, oils from different plants like fennel is there. It is also used to extract out proteins from different materials like from peanut as peanut proteins, from rapeseeds, from rice bran and soya protein. From soya we extract out soya proteins. In food industry, enzyme assisted extraction is also used to prepare probiotics because these enzymes help in disintegrating the fibers. They help in reducing the structural integrity of the dietary fibers, which can be used as probiotics. These enzymes also help in improving the rheological properties of the dough by hydrolyzing the cell walls of the grains, and such kind of good quality doughs are used in bakery industry. This technique is used to extract out oil from different plant material like olive oil from olives, any kind of fixed oil also from different types of seeds, to extract out antioxidative compounds or phenolic compounds from different raw materials. This technique also helps in enhancing the yield of the juices from the fruits. Specifically, pectinases are used in fruit juice industry. Enzyme assisted extraction is also used in wine industry to enhance the maceration of the grapefruit, to enhance the extraction of the color from the grapes, to enhance the extraction of phenolic compounds from the grapes, which enhances the taste as well as the quality of the wine. It is also used in paper industry to improve the pulp, the quality of the paper pulp, by removing the lignocellulosic components from the paper material. So friends, this was all about enzyme assisted extraction, starting from the composition of the plant cell wall to various hyphenations to the applications of the enzyme assisted extraction. Thank you.